Good afternoon, everyone, and welcome. My name is Vivian Hanrahan, and I am the webinar coordinator for the HRB-TMRN, and you're very welcome to our webinar series. Today, we'd like to welcome everyone, and are especially delighted to welcome Professor Jamie Kirkham. Jamie's joining us today to give his webinar, which is entitled A Random Walkthrough Outcome-Based Research in Health for Healthcare. So quick bio on Jamie. He is a professor of biostatistics at the University of Manchester, having previously held a post for 13 years in the University of Liverpool. He joined Manchester in June 2019, and his role focuses on statistical and methodological research for clinical trials and systematic reviews. He has expertise in outcomes-based research, particularly in the field of core outcome development and outcome reporting in clinical trials. He's also interested in study misconduct, associated biases, and the potential impact of these failings on evidence-based decision-making when important outcomes from studies are not measured or selectively reported. Jamie's also statistics editor for the BMJ and has an interest in journalology, which is the publication science. So you're very welcome today, Jamie.
Thank you.
Okay, just before I hand the webinar over to you, I'd just like to draw everyone's attention to the Q&A box at the bottom of your screen. If you hover over that, you should be able to type in your questions throughout the presentation, and then I can put them to Jamie at the end if that's all right. So without further ado, I'll hand over to you, Jamie.
Okay, thank you very much for this kind invitation. Okay, so throughout the last 15 years of my career, I've been working loosely on outcomes-based research. And in this webinar today, I'd like to take you through the journey of some of that research that I've been involved in over those last 15 years. And that's really where the title of this talk stems from. The Random Walk is really taking you through all the aspects of the research that I've been involved in. Not necessarily the order that research was done, but I think one that tells a nice story that describes the importance of outcomes-based research in healthcare. Okay, so I think first and foremost, it's really important to identify why outcomes are important within research studies. Interventions are compared in randomized controlled trials by measuring differences in patient outcomes between those groups. In order to do that, we need outcomes that are relevant to all relevant stakeholder groups. So selection of the appropriate outcomes is really crucial. And I think one of those things that's really important here is that there are many new methodological aspects of clinical trial methodology that's being developed. For example, there are more efficient adaptive designs where there's lots of research into the recruitment and the retention of patients within trials. But at the end of the day, if the outcomes that have been important are not relevant, then we must question the how useful those trials might be. Back in 1993, Peter Tugwell said that clinical trials are only as credible as their outcomes, and I think this is really important and current today, but I would also think this extends out to things like systematic reviews and clinical guidelines. So I just really want to focus on why outcomes might be important in a number of different frameworks that already exist, and one of those is in research waste. Back in 2019, a series of papers were published in the Lancet series focusing on research waste, and they all concentrated on different stages of the research process. So right from the very beginning, when we're asking questions about the types of questions that should be answered, are they high priority questions? For example, do we really need to answer those questions in the first place, or have they already been satisfactorily answered by other researchers? Right through to trial conduct, thinking about publishing the final study findings. This is really important. In many cases, the publication is the only tangible evidence that the study was ever done in the first place. So it's really important that we publish our research findings. And in the final section, thinking about when we do publish our study findings, we make sure that the findings are complete, transparent, and can be replicated. So the areas of research that I've mostly focused on is the extremes. So thinking about making sure at the beginning of the process, the outcomes that are measured are important for all relevant stakeholder groups, and that may include healthcare professionals, patients, amongst other important stakeholder groups. And then right at the end of the process, thinking about, once you've decided on those outcomes, making sure that they're fully reported in an understandable way. I've also focused a little bit more recently on fully accessible research and open science. So I'd like to spend a little bit of time talking about that as well, because I think that's really, really important. And I think this is something that we need to concentrate more on in the future. So packaging all that together, this contributes to research waste, where there's these inefficiencies in the research process. And one of the overarching conclusions to those Lancet series of papers was that tens of billions of dollars of investment in research are lost every year because of correctable problems. And of course, there are many initiatives out there that are trying to reduce this amount of research waste. I will talk about some of them today, and as I've been fortunate enough to be involved in some of those. So one of those initiatives is the MRC NIHR Trials Methodology Research Partnership, or the TMRP. So really at the central sort of engine room to this partnership is its working groups, and there are a number of different working groups that focus on different aspects of clinical trial methodology. So the ones that I'm most interested in are the ones highlighted in red, so the one on global health and the one in outcomes. And there are specific target research areas within those groups. So one of those within the outcomes groups is statistical analysis plans and outcome reporting bias. So I'm going to tell you a little bit about the research I've been doing in that particular domain. Also, the widening participation in core outcome set development. So alongside colleagues within Liverpool, I've developed a number of guidelines to help people develop core outcome sets, but also actively supervising core outcome set projects. So I can mention some of those. And within global health, one of the key priorities is about the appropriate choice of outcomes in low middle income country settings. And this is where I've started to invest a little bit of interest and research within this particular area. So I will focus on that towards the end of this presentation. And as you can see, within those working groups, there is clear cross-cutting themes that go alongside each of those working groups, and this is a good example of that. So choosing outcomes and low middle income settings clearly cuts across global health and the outcomes working groups. So this is an example of a particular problem that we see with outcomes, and those of you who are on the call who are systematic viewers may see this on a regular basis. So this is looking at trials, drug trials for rheumatoid arthritis. So on the column on the left-hand side, we have a number of different studies that have looked at trials of rheumatoid arthritis. And across the top, we have a group of outcomes that are considered really important for rheumatoid arthritis. So they aim to measure disease activity, how a patient might be feeling, and a measure of damage of the joints. So this is actually a core outcome set, but I'll focus a bit more on that towards a bit later in the presentation. And what we see here is that the ticks represent the studies that have measured those particular outcomes, and where there's gaps, that's where these studies have not measured those outcomes. So these gaps can occur for potentially two different reasons. First of all, it might be just a missed opportunity to measure a really important outcome. Alternatively, there might be something more sinister going on here, and maybe they did measure the outcome and perhaps they didn't report it because of some knowledge of the results that they found. And this is really outcome reporting bias, which is where I'd like to focus a little bit of our time. Regardless of the reason though, both these reasons contribute to research waste. Okay, so when I first moved to Liverpool as a postdoc, one of the first projects that I was involved in was outcome reporting bias in trials. So this had both qualitative and quantitative components. And this is one of the highlight results from the qualitative research. And I'll give you a chance to read that quote, but what really is going on here is that we can see that actually some researchers were clearly not reporting certain outcomes because of some knowledge of the findings. And in this particular scenario, it's because the results didn't show anything particularly interesting, or the results were not statistically significant. And this can contribute to a bias within the evidence base. As well as this qualitative research, we also conducted some quantitative research, but I just firstly wanted to show you this trial flow diagram. And this really highlights where we can get these particular problems and how they occur. So let's assume that a trial starts. Once a trial starts, data is collected, some analysis may be done. If that analysis didn't get submitted, or it did get submitted but not accepted for publication, this is where we can get publication bias. So the whole study is not published. And again, this is really, really an important concept. And within my slides about open science, I will talk a little bit more about publication bias. But what say the study did get published, but they only published a subset of the outcomes that were pre-planned. And this is where we get the problem of outcome reporting bias. And the bias mechanism is really acting if the decision not to report those outcomes is based on knowledge of the results or the significance of those. So where we'd really like to be is in that bottom right quadrant, where the study is published and all the pre-planned outcomes are reported in full. So there's a number, I've talked a little bit about the quality of evidence. Here's some of the quantitative evidence. So this was a systematic review that was done looking at cohorts, following up cohorts of studies that had followed those studies from inception right through final reporting. And we can see across those studies, there was much more chance that statistically significant outcomes were reported than their non-statistically significant counterparts. Also, that the studies found that looking at reports of versus protocols, quite a high percentage of primary outcomes were changed, newly introduced, or omitted. So there's a really strong signals that the reason for not reporting may be based on some knowledge of those findings. So throughout my first research associate project, we developed a classification system for trying to identify sources of bias as a result of not reporting certain outcomes by looking at lots and lots of different trial reports. And we assigned those high and low risk, and that the high risk was really contributable to those non-significant findings. So this was done in 2010, and I think what was really the positive aspect of this study is that we could quite well identify whether the risk was high or low just by looking at the actual trial reports. There's a lot of emphasis on trying to look at alternative sources, so protocols, trial registry entries, etc. But as we all know, those are not always available. So sometimes we do have to try and make the assessments just on what we know by looking at the trial reports, and we were actually able to do that quite well in this particular situation. So within that particular study, so we looked at cohorts of trials within Cochrane systematic reviews. So Cochrane reviews are known to be the highest form of evidence for making clinical decisions, but we actually find in over a third of the reviews that we looked at, there was suspicion of outcome reporting bias on at least one primary outcome. As I already alluded to, based on our assessments, we'll actually be able to identify the bias in a high proportion of cases. So what we actually did, we actually followed up trialists and asked them if they actually measured the outcome that wasn't reported within those reports. And they were able to confirm whether they were measured or reported or not. And we can see that we can do that a high percentage of time. So actually our classification system worked really well there. And I think this is, as I said, I think this is particularly useful because in many cases, you're unable to obtain the other source documents to make these assessments. So what impact does this type of bias have on the evidence base? Well, this is an example of a funnel plot. So the one on the left demonstrates when outcome reporting bias or publication bias for that matter is not impacting on the systematic review conclusions. And on the right hand side, we can see where it is having some impact. So it's those smaller non-significant studies that are missing from those meta-analyses, and we can see it has a tendency to overestimate the treatment effect. We also applied some sensitivity type analysis in that particular study to significant meta-analyses, and we found that actually once we made our adjustments, 8 of those would have not have ranged statistically significant, and 11 would have overestimated the treatment effect by more than 20%. So we can see, this can be a potentially quite a serious problem going forward in terms of making clinical decisions. So what are the solutions to outcome reporting bias? Well, of course, there are non-statistical solutions and there are statistical solutions. Of course, as what the Cochrane guidelines will point out, is that you need to try and contact trialists to get the missing outcome data that you need to complete your review. I think there's been varying degrees of success with this. However, there's a really nice article that that was published in the BMJ, and it gives a nice overview of where the types of sources that you can look to try and obtain this missing data. So ranging from looking at trial registries, results database, regulatory online databases, data request, trial and sponsor contract, conference abstracts, and online search. So this can be quite time-consuming processes, and I think, as I alluded to, there's a varying degree of success from using these methodologies. Another non-statistical solution is the idea of a core outcome set. I've been involved in core outcome set development for quite a while now. So they are an agreed standard size set of the most important outcomes. They can be disease or condition specific. It can be all treatment types or of a particular intervention. So as an example, the outcomes I showed for rheumatoid arthritis, so that's the disease and condition, and that core outcome set is mostly for drug trials. They can focus on the benefits and the harms, and they are a minimum set of outcomes, so there is no restriction. So it's basically a core outcome set of the outcomes that we'd like researchers to use for those particular conditions for which the scope has been defined. But if researchers want to measure other outcomes as well, they're more than welcome to collect other outcomes. Most of them to date focus on effectiveness trials, but there's a growing interest and developing those for routine clinical practice. So this is a really good example of the impact that core outcome sets can have. So this is actually looking at the uptake of the rheumatoid arthritis core outcome set over time using trials that have been conducted with this particular condition. So there's lots and lots of trials. I think this evaluation includes over 300. And we can see that over time, the uptake of the core outcome set for rheumatoid arthritis has reached nearly or just over 80%. So the core outcome set was developed in 1994, and at 2020, I say we're reaching just nearly over 80%. So if there is any real criticism here, it's perhaps that it's probably taken too long to get this point. So it's been over 20 years to get to that particular point. So I think there are further things we can do to try and increase the rate of this uptake. Anyway, this is a really good example of how useful core outcome sets can be. However, we also need to be cautious because our assessments actually were made up of those where they planned to measure those core outcomes. But we did another evaluation with Giacomo, who was one of my PhD students, and he looked at the uptake of the core outcome set for rheumatoid arthritis in reviews, and he found in all 21 reviews, there was missing data on at least one of those core 8 outcomes. So what's going on here? Well, this is a particular example from a Cochrane review, and this seems to be going pretty well. So again, like the matrix before, we have the studies on the left-hand side and this core outcome set in the top row. We can see most of these trials do seem to be measuring these outcomes. We can ignore radiological damage for this point because actually there's some condition on this outcome that it only needs to be measured in longer term trials. So we'll ignore that for now. But this seems to be going quite well. But what was actually reported in the trial reports? Actually, none of the studies actually reported on all the core outcomes. So what was actually going on here? Well, it was actually this to do with composite outcomes. And there is actually a core composite outcome for rheumatoid arthritis that encompasses all the individual outcomes within the core outcome set. And actually the regulatory guidelines that was put together, they said that you need to measure a measure of disease activity for which one of these core composite outcomes is, but you should also report all the endpoints and change in the single variables included in the core outcome set. So what these trialists have been doing, they've measured and they've reported on the composite outcome, but they've not actually been reporting on the individual outcomes within the core outcome set. So again, for me, this constitutes research waste. Knowledge of the individual results to those individual outcomes is really important, because some of them reflect what's more important to patients than perhaps the composite outcome would do so. So I actually wanted to investigate this a bit further because this slightly annoyed me. And of all those trials within that core outcome set uptake evaluation, so there's 341, 96 of them plan to measure the composite outcome. But only 16% of those 55 plan to report on the individual core outcomes separately. So despite the regulatory guidelines on what you should measure and report within these trials, clearly in the majority, I've just gone in darkness. Never mind, I'll carry on regardless. So 50 plan to report on the individual core outcomes separately. So clearly the researchers are not following this regulatory guidance on what they should measure and report within clinical trials. Sorry, somebody's just going to see to the lighting.
Okay, Jamie?
Yes, okay, we're back on. I've got power again.
Great.
Okay, thank you. So yeah, I actually put this to one of the editors, the chief editor of one of the main journals in rheumatology. Most journals actually endorse this particular guidance. And in fact, the chief editor was on that list of authors there. And we had lots of debate going to and from, and he was of the opinion that, well, if trialists only measure and report on the composite outcome, they're actually utilizing the core outcome set in a meaningful way. I still disagree with this, and eventually I did get my way, and we published a nice letter within this particular journal, and we're trying to push forward to make sure that going forward, that all these individual core outcomes are measured as well as the composite outcome. So whose fault is poor reporting? Well, I think poor reporting indicates a collective failure of authors, peer reviewers, and editors on a massive scale. Research authors may not know what information to include in a research report, and editors may not know what information should be included. So what information can we give to authors and editors? Well, clearly, most of the important information is reporting guidelines, but as we've already seen, even though journals are supposed to endorse them, even though authors are supposed to follow them, clearly that's not the case. So I think we just need to provide more education and more screening checks on what gets put forward for publication. So as in my introduction, it was mentioned that I am a statistical editor for the BMJ. The BMJ is very research orientated, and they do implement many things to try and prevent things like outcome reporting bias. For example, for publication, it's a requirement that the trial is prospectively registered, so we've got good insight into what they plan to measure at the very beginning of the process. They also require the protocols to be submitted with any trial publication, and they also need to declare a transparency declaration that what's reported in the trial report is a true reflection of what they actually did within that study. Despite this, there are still some inconsistencies, and we can still see that some pre-specified outcomes are not reported and outcomes are newly introduced. So we did this particular study. We looked at 275 trials, and we looked at a sample of those because only 254 were actually rejected by the journal at the early phase, and only 21 were accepted. for publication. All of them had protocols, so they definitely adhered to that particular constraint. And we actually used their manuscript tracking system to see if any discrepancies in outcomes were picked up by editors, or were they picked up in the peer review process. And we found in the majority of cases they were, but as I said, there were still percentages that were where there was some irregularity. Within the responses to those peer review comments, authors said there was space limitation, outcomes were not yet analysed in full or reported elsewhere, or there was some error within the manuscript or the registration of that particular trial. What I would say is that most of those reasons that they described didn't really, in my opinion, suggest that there was any bias going on there. I think they were legitimate. If we have a reasonable degree of trust in what they were saying, I think they were legitimate reasons. And we see lots of these evaluations all the time where people look at these types of things with lots of different journals. And the BMJ generally comes out quite on top with those evaluations. So as I said, they do have the requirements and they do actively check these things. So I think they do a reasonably good job here. But I think we need more wider participation in this sort of activity across different journals. So which is the bigger problem? Is it outcome reporting bias or is it publication bias? Well, within that systematic review I talked about, we actually got the actual data from one of those cohort studies that I talked about. And we actually looked at this data in more granular form, in more detail. And one of them was looking at findings from a cohort of trials submitted to a German research ethics committee. And we actually extracted from the 308 studies in that review, of which 167 were published, all the outcome data from those studies, and we looked at all the different sample sizes, so planned sample sizes and final sample sizes for those that were published, and we looked at the amount of proportions of reporting or missing at the individual participant level, and what we actually found is that missing data attributed to selective reporting was only 4%, and the proportion of missing data from unpublished studies or publication bias was actually 15%. So actually publication bias was the larger of the two problems. But we also found within that study that there was lots of partial reported data. So essentially my definition of partial reported is that they'd actually reported some data on the outcome, but it wouldn't have been sufficient enough to include in any sort of review meta-analysis. So again, this adds to that problem. It's part of the research waste problem. So actually in total, the proportion of missing data that's may not be that useful, particularly for inclusion in systematic reviews, was actually as high as 53%. So I've talked a little bit about my involvement in journals, and I think, and it was also mentioned, I have an interest in publication science. So one project that I'm particularly leading at the moment is to do a systematic review of all preprint servers that have scope that's both medical and those in health and life sciences. And there is actually nearly 50 of those which we were involved, and we've collected lots of information from the policies of those preprint servers involving all the preprint server owners. So for those who are not familiar with preprints, essentially you can actually publish your findings immediately. So you submit your manuscript and your paper was then published within a few days, but there is no peer review process. So this means the dissemination research is much more rapid. It improves the openness, so you can publish these free of charge, so there's no charge to the authors. And actually, contrary to belief, even though there is no peer review, we found from our review that the majority of these preprint servers do have quite rigorous screening processes. So there is a process whereby the content is checked to see if it would be harmful if it was made available to the public domain. And contrary again to belief, even if you pre-printed your work, there is lots and lots of options for authors to go to any other journal of their choice to later get peer review of their manuscript. So I think this is really important in terms of trying to reduce the impact of publication bias. So here at the HRB, where I'm doing this webinar, they actually have their own preprint server, and I'll use preprint loosely here because they use the F1000 platform, which means that they look to seek post-publication peer review, but it certainly has preprint functionality, and they are actively using this preprint server. And I can see, I did a quick search on there, and they have nearly 100 articles on there now, and they're using this mostly to publish all their protocols for the studies they're involved in. And I think that's a really good use of these preprint servers, because quite often with protocols, we don't really get much opportunity or time to wait for the peer review, because we kind of need to start our project quicker than the four or 5 months it may take to get that peer review. So I think this is a really good way to post protocols. So we've got some transparency and some early acknowledgement that the study's being done. So I've just screenshot one of those, and this is because this is a particular project they pre-printed that's looking at core outcomes and attitudes of questionnaires to completion of response rates within trials as a study within a trial. So just changing and going back to reporting bias again, as well as looking at efficacy outcomes, we also looked at the problem of outcome reporting bias within harm outcomes, within Cochrane reviews again, but also non-Cochrane reviews. And as you can imagine, the actual problem is even worse for harm outcomes. And specific harm outcomes reported in only 38% of Cochrane reviews. Overall, 3/4 of the primary studies in the Cochrane reviews did not report the relevant review primary harm outcome, or did not report it in full. So this is quite high percentages. Again, jumping around a little bit more. So I've really focused only on the problem in terms of reporting bias in clinical trials. But we also find with this Cochrane methodology review that the problem also exists in selective reporting within reviews themselves. So looking, again, studies that have looked at discrepancies and outcomes that were listed in Cochrane protocols and those in the final review differed somewhat. And again, the impact can be the same as those within trials. So as well as those sort of non-statistical solutions to outcome reporting bias, so looking or trying to obtain the missing outcome data or core outcome sets, we've also looked at some statistical solutions or sensitivity analyses. And over time, these methods have developed, and we've I think we've settled on a method that we think is the best at this current time going forward. It's an explicit modeling technique that was developed in collaboration with John Kopas. So again, this is an example. So it's a sensitivity analysis. The application is in epilepsy. So we have this similar matrix. So we have the studies on the left-hand side. We have all the outcomes that were looked at within that particular review. And notably this time, there's focus on both benefit outcomes and harm outcomes. So the tick represents where those studies measured and reported those outcomes, and the crosses represent those that didn't measure those outcomes, and the circles represent where it was only partially reported. For the purposes of this class presentation, we'll ignore the letters, but the letters represent the level of risk of bias according to the classification systems that we developed, which I mentioned at the very beginning of this presentation. So actually in this example, the primary benefit outcome is actually reported in most of the studies, but there's clearly lots of missing data on the harm outcomes. So we developed this modeling technique as a sensitivity analysis, and the actual mechanism for harms actually differs for benefit outcomes. So with benefit outcomes, we expect the bias to occur as a result of non-significant outcomes, but with the harm outcomes, this actually may get reversed because you may choose not to report on the harm outcome if you observe more harm in one particular treatment arm. So this was the unadjusted results were the analysis for all those outcomes as reported within that Cochrane systematic review, and the adjusted outcomes are those from this sensitivity approach. And we can see for the benefit outcomes where there's very little missing data, there's very little adjustment in the estimates between the unadjusted and the adjusted. But for some of the harm outcomes where there was more missing data, we can see more severe changes in the estimates. And actually the ones that I've highlighted is where the results, where the significance was overturned. We perhaps shouldn't focus too much on the significance. It's really about whether that difference actually makes is clinically importantly different. But for the purpose of this presentation, I thought it was just quite nice to see where there was some change in significance. As well as thinking about all the methods that we've developed for reporting bias, it was also important to do some educational work to make sure that all the tools that we've made available can be used by non-methodologists. So that might be that matrix system that I've showed. It might be the classification system for detecting the level of risk of reporting bias within individual studies, or it might be that particular sensitivity approach. And as a part of this educational initiative, we run lots of different workshops at the time, but we also produce this educational article on outcome reporting bias, which goes through the steps and the processes that systematic reviewers can find to first identify and try and address this problem within their particular reviews. And we also developed a website as well that includes all the resources, all the tools available that can be easily used and downloaded and included in systematic reviews, as well as providing a web-based platform for trying to apply the adjustment method without the need for any knowledge on all the statistical methodology behind that particular approach. So again, I think this is really important actually. So it's in the context of research waste. If we're going to do this methodological research, if people are then not going to use it in a meaningful way, then that can also be considered as wasteful. So I think this is one of the, again, quite important going forward, that we make sure that users of our research or methodological research, it can be used easily and it's accessible to them. What about the larger impact on things? Well, many of you may, if you're systematic reviewers, may have seen these two tools. So this is the new risk of bias tools used for the Cochrane collaboration. So the first one, Robin's Eye, is for non-randomized studies, and the second one is for clinical trials, randomized trials, risk of bias 2. So within those two tools, there's a particular domain looking at selective reporting of outcomes. And very much that work that we did on Orbit is fed into those two risk of bias tools. So we anticipate that those will be widely used. If you've used those tools, you probably either love them or hate them. But really, what I can say is being involved in the development of those, the idea behind them is that it gets you to think more about the trial as a whole, what was done, and the signaling questions try and aim to get you to a more informed decision about the risk of bias of that particular study for each domain, rather than just thinking that the risk of bias might be unclear. So maybe some of you have got some experience with using those. And there's actually another tool that's been developed as well. So within the randomized trials risk of bias tool, it really focuses on the selective analysis reporting of a particular result that's been observed within that paper. The selective non-reporting, which is more down the line of the outcome reporting bias work we did, is going to be in a new tool that's going to be called RobMe. Okay. We've also engaged, moving forward and jumping around a little bit more on this journey, we've looked at where core outcome sets have been developed or are ongoing across the different review groups within within the different review groups within Cochrane. So we can see this is quite highly varied. So the most active core outcome set development group appears to be musculoskeletal, and that's clearly because of the OMERAT group that have developed lots of core outcome sets for different musculoskeletal conditions. The rheumatoid arthritis one, which I mentioned, is one of those. But perhaps one of the growing areas of interest, is which we've had some discussions about this morning, is in pregnancy and childbirth. And we can see that there's a significant number of ongoing core outcome sets within this particular field of research. As part of that research, we did some surveys with Cochrane coordinating editors, and we did this a number of years ago, but we've also done it more recently. And in the current survey, 86% of those coordinators now think that core outcome sets should be used routinely within summary findings tables within their reviews. There was clearly different review group policies on outcome selection, but since we did the first survey, there's been more of a shift towards actually a review group policy and a negotiation with authors on the choice of outcomes to include in their review, and this may be informed by available core outcome sets. Previously, authors were given more of a choice about which outcomes to include within their reviews. But the three main challenges of core outcome set development still remained: the development of the core outcome set, the scope of the core outcome set. So some review groups felt their review group was quite broad and found it quite difficult to encompass core outcome sets, and actually persuading authors and trials to implement those core outcome sets. And in more recent research that I wanted to mention, I'm quite interested in thinking about the challenges of incorporating those from low middle income country settings within core outcome set development. I'm actually involved in a research project funded by the University of Manchester, also involving Declan from the HRB, and we're actually going to be focusing on pregnancy and childbirth. So we have already an existing network from 6 sub-Saharan African countries, and we're going to go and we're going to try and find out more about their healthcare systems and how to include participants from these particular settings within the core outcome set development project. We're focusing on pregnancy and childbirth. As I've already said, this is a growing area of core outcome set development, but I think it's actually really important in this particular setting. Most core outcome sets within pregnancy and childbirth focus on complications of pregnancy, essentially. And actually, for many of these complications, the prevalence of these complications actually, in some cases, 100-fold higher in these low middle income settings than any other way in the world. So I think it's really important that we do try and engage participants from these particular areas so they get some form of opinion on important outcomes for these particular conditions. And my final sort of part of the journey is that I focus mostly on outcomes, but there's also a different form of selective reporting, and that's selective reporting of analyses. So that's where we have some data on particular outcomes, and we may choose to analyze the data in different ways to potentially obtain a more favorable result. So I've already loosely talked about composite outcomes, or use of those, but we've also seen other things in the literature as well. So dichotomizing with continuous outcomes is quite popular. Different ways of dealing with missing data, also different statistical analyses that we may perform on our data to make results more favorable. So as well as outcomes, we also need to be a little bit cautious of changes in analysis plans. And this is something I think that we need to think about more in the future. Okay, that really brings me to the end of my webinar. I've talked about lots of different aspects of outcomes work that I've been involved in, and thanks for listening, and I'll look forward to any of your questions.
Thanks so much, Jamie. That was really, really good. Thank you. Loads of information to take in there. We've had a good few questions as well, if that's okay. So hopefully we'll have enough time to cover them all. Our first question comes in, and it's, let's see, It says, evidently you've shown that deciding on important outcomes in research is crucial to avoid research waste. But have you any, how have the results of this research been taken up by clinical trialists? So how do they react to your, to addressing the core outcome sets?
I think it's, I think it's an important question, and I think it's something we would probably need to do more work on. I think in the example I showed on rheumatoid arthritis, so we can see that the increase is nearly just above 80% now, which is taking us quite a long time, as I said, in my opinion. And I think the challenge is trying to get that beyond. You probably saw from that particular graph that the uptake line has started to plateau off a little bit. And I think we need to have an extra push or intervention, I think, to try and get that even higher now. I think that's one of the biggest challenges. I think there will always be some resentment. I think, naturally, some people just don't like to follow guidance, and they will just like to do their own particular thing, and they will never measure that core outcome set for reasons of their own. Maybe they're involved in the process, and they didn't agree with that particular process or agree with the outcomes that came out, and they choose not to measure them. So I think we will really struggle to get to 100%, but I think we need to do perhaps more qualitative research to understand the reasons for not reporting on certain outcomes.
Yeah, absolutely. Just slightly on that tone as well, Declan sent in a question, and he said core outcome sets are likely to go out of date as clinical practice changes and evolves. What are your thoughts on how core outcome sets can be updated efficiently?
I think there is this. I think this is going to be a problem for the future. I think core outcome sets will potentially need updating. I also think that from what we know is that some core outcome sets have not been developed, perhaps using the rigorous methods that we've outlined in some of our guidance. So for example, perhaps they've not included patients. So I think some core outcome sets may need to be updated to include their particular views. I think in terms of making it more efficient, I don't think we can keep going right back to the start of the process and reevaluating things right from the beginning to generating lists of outcomes to doing Delphi type processes. But what I think we can do, I think we can identify potential for doing some sort of top-up type research, so we can sort of supplement existing research in a much more efficient way to try and bring those core outcome sets up to standard or update them to current day practice.
Yeah. Okay, another question's come in here from Joseph. How can we set the priorities of outcomes when designing primary research using the outcomes from core outcome sets, especially when there are concerns around sample size?
Yeah, I think that is an issue, and I think it's about involving all those all relevant stakeholders within the core outcome set development process. And an example of might be that is actual funders of trials. So there's a clear understanding of what's actually achievable when we're deciding on our core outcome set. So actually, the rheumatoid arthritis example was a good example of that. So you probably saw that the outcome that was least reported was that of radiological damage. And that's because it's quite an expensive outcome to measure. It takes quite a lot of resource. It requires specialist types of assessments and experts to essentially review the radiographs for that particular outcome. So I think we need to make more careful attention to that, really.
Okay. So another question here. Sorry, I'm just a bit distracted there. I was coming in on different chat lines. It says, how do we as researchers promote transparency among our wider research group and act as advocates to combat this problem? So again, that's the trialist uptaking and the core outcome sets.
It's something I mentioned last week in a talk I did. Really, I think it's about trying to almost. I think we're quite conscious that we continue need to do novel and new research all the time, and I think maybe we just need to step back away from that sometimes, and we need to promote the existing research that we're doing to the relevant communities, and those really educational initiatives that I really talked about. I think it's really important to make sure that our research is more widely accessible and reaches out to the relevant communities for which they were developed for.
Yeah, okay, that totally makes sense. And then our last question that's come in, it says, how as researchers do we ensure that we don't contribute to the missing data going forward? Do you have any tips for being that's more about transparency, about being as transparent as possible within the word limits count of the journal? So
to be honest, I think that's a non-existent problem these days. Most journals are open to, much more relaxed on word constraints, but also, it's almost endless because you can put as much information as you like in supplementary material. And also, I would encourage people to use data repositories as well, as such things like Zenodo for uploading data sets for their particular studies. That's something I've started to do more frequently. Yeah, I just think it's a non-problem these days.
Yeah, okay, well, that's brilliant. Thanks so much, Jamie. That was really, really informative. I think we had a huge audience today, so I think everyone's got a lot out of that, and it's been really good because I am a complete novice on core outcome sets, so it was really understandable even to me. So thanks so much, that was great. I just want to, before we finish up, just to turn the attention of everyone listening to a couple of things that are coming up. On the 27th of February, there's another webinar given by the Common Initiative, so a bit more information on core outcome sets, and it's titled No Choice of Outcomes About Us Without Us. So that will be at 4 p.m. on the 27th of February. Then on the 9th of March, there's a study day coming up in University College Cork, and it's with regards to registry-based randomized trials from concept to practice. And then lastly, our next HRB-TMRN webinar will be on Tuesday, 21st of June, and that's with Dr. Stephen Walter, and he is going to talk about the implications for estimating treatment effects when trials stop early, either for apparent benefit or futility. So that would be our next webinar. Thanks everyone for joining us, and thanks especially Jamie. That's really great that you've taken your time out of your day to have a chat with us. So all the best, and hopefully we'll see you at the next webinar.
Okay, thank you very much.
Thank you. Bye-bye.